when we talk about disease states, the name is assigned based on how a physician would diagnose a disorder based on what they see in the routine diagnostic test. Today, that diagnosis doesn't begin with genetics because genetics is a more recent discovery. So while cystic fibrosis sounds like one disease, there's hundreds of different genetic variations that affect this one essential protein, the chloride channel. And depending on what type of genetic alteration there is, different drugs or different treatment approaches make sense. We focus in on nonsense mutation, and this is a change in the gene that inhibits or prevents the protein from being made. So our drug stabilizes the ribosome so it can help individuals make that protein, the CFTR again. Yeah, we're very excited about ELX-02's potential in cystic fibrosis because next week we'll be at the European Cystic Fibrosis Conference in Belgrade, Serbia, presenting 2 oral presentations on early stage experiments with ELX-02. The first experiments are about human bronchial epithelial cells. Now, these have been used for years to screen drugs for cystic fibrosis. We'll be sharing the latest results. But also the organoids that I mentioned a moment ago. These are actual stem cells from patients with cystic fibrosis, and we'll be sharing the results of how those cells responded to ELX-02. We think the data are very exciting and really looking forward to our meeting next week. Yeah, we're submitting for regulatory approval to open a phase 2 trial this year, and we're right now waiting to hear back from the regulators that we have the green light to get started. But we do anticipate this year beginning our phase 2 trial in cystic fibrosis in Europe. To go. When we think about cystic fibrosis and different genetic changes in the gene that codes for the protein, remember we make messenger RNA, and then the messenger RNA serves as a template for making proteins. And there's a whole series of different changes that can occur in the messenger RNA that impact the protein. So we focus on messenger RNA changes where there's an accidental stop codon, part that blocks the protein production. And our drug interacts with the ribosome to allow an amino acid to go into the premature stop codon so the protein can be made. There's over 1,800 human diseases where premature stop codons prevent the production of essential proteins.